Recording in progress.
Good afternoon, friends of CAFÉ CON. Welcome you to our channel of scientific conferences of the Real Sociedad Económica Extremeña de Amigos del País in Badajoz. We are happy and proud to be able to offer you this conference in English for the first time in the 205 years of life of our society. This is a very historic moment in our society. And this afternoon, we are going to talk about a very interesting topic with the Professor Alessandro Feis from the University of Florence in Italy. Good afternoon, Mr. Feis. How are you?
I'm fine, thank you. Good afternoon to everybody.
And you are going to tell us about the photoacoustic effect and its interesting and novel applications in the field of biology and medicine, isn't it? Yes, you are going to tell, yes. And you are a professor, associate professor in the Department of Chemistry at in the University of Florence. Well, you were born in Florence, there in Italy, and you are living at home. And you graduate in this university in 1985. You received your PhD in chemistry from the University of Padua in Italy in 1990 after an experimental work of nonlinear spectroscopy. You work as a postdoctoral fellow at the University of Mainz in Germany, at the former Max Planck Institute for Radiation Chemistry in Mulheim, Ruhr in Germany, where you learn photoacoustic spectroscopy with the Professor Sylvia Braslowski group. Really difficult names. I have to apologize. I tried to say it right. And then you joined. Yes, it was right. Yes. Then you joined the Department of Chemistry of the University of Florence in 1996, where you collaborated with Professor Julieta Smulevich. After that, you spent a sabbatical year in the Institute of Structure of Matter of the CAFÉ in Madrid, and in 2012, in collaboration with Dr. Santiago Sanchez Cortés. And then you visited the Faculty of Chemistry of the University of Duisburg-Essen in Germany in 2016 and 2017. Your main research interests are in the fields of Raman spectroscopy method. You were do you remember you were in our conference about Raman spectroscopy? Right. And in optoacoustic studies of molecules and nanoparticles. All this kind of matter is the topic that concerns us this afternoon. To discuss this issue and establish an interesting dialogue after your conference, today we are joined by three leading experts in this field that's as far from Chile, Brazil, and Madrid. And we are connecting Badajoz, Florence, Brazil, Chile, and Madrid. And first of all, in first place, we have to say hello to Mrs. Natalia Inostroza Pino. Hello, Mrs. Natalia, how are you?
Hello. Fine, thank you.
And where exactly are you in Chile? Which city, which town?
Santiago.
In Santiago,
yeah.
In the capital. Yeah. Okay, yeah. Really nice place. I have a friend in Chile, it's really nice place. You are a PhD in molecular physical chemistry at the Universidad Andrés Bello in Chile. With an amazing curriculum full of courses and research papers. And you are the main researcher in a very interesting project called Astrochemistry in Terrestrial Complex Organic Molecules through gas-grain and grain-grain collisions. In that international project, collaboration project of CONICIT in the Agency of Chile, and in collaboration with NASA and Max Planck. And your scientific interests are astrochemistry, structural and spectroscopy properties of relevant systems, quantum chemical determination of structural and electronic properties of pollutant and greenhouse effect in the atmosphere. Really actual subject is
hot topic.
hot topic,
yes.
the greenhouse effect
Yes.
and all these kind of
Yes.
things. And then light absorption process, inorganic dye synthesized solar cell. A lot of really high trending topic. That's good afternoon, Mrs.
Thank you.
We are going to
you
say
very
hello to the other debaters, and then you can ask to Professor Alessandro. And it's a pleasure to have a second participant, is Professor Leonardo Farini from Brazil. Hello, Mr. Farini. Where exactly are you in Brazil?
Hola, I'm in Florianópolis, in the south of Brazil.
Florianópolis, really interesting place. Leonardo is professor at the Department of Physics in the Universidad Federal de Santa Catarina. He obtained his PhD in material science at the Universidad Estadual Paulista, and his expertise in surface and haze Raman scattering applied to detect environmental contaminants. Part of his PhD was done at Instituto de Estructura de la Materia, CESYC in Madrid, and University of Victoria in Victoria, Canada. Also, he did his postdoctoral period at Universidad de Vigo in Vigo, Spain, and his area of interest is vibrational spectroscopy, optical properties, surface characterization, nanoparticle synthesis, and thin films. Wow, Mr. Farini, really amazing. We will you will have a lot of questions for Professor Feis. And finally, yes, and finally we have a friend in Madrid.
Hello.
And hello, Mrs. Mari Vega. She is our old friend of CAFÉ CON. She participated in the conference of Raman spectroscopy too. And Mrs. Maria de la Vega Cañamares Arribas is a senior scientist at the Institute of the Structure of Matter of the CAFÉ. Is graduated in chemistry and also obtained the PhD in chemistry, both from the Complutense University of Madrid. During his doctorate, she worked on the application of surface and haze Raman spectroscopy to the study of natural colorants of interest in cultural heritage. She did her postdoctoral stay in the Department of Scientific Research in the Metropolitan Museum of New York and in the Department of Chemistry of the City College of New York. She worked in the application of this source spectroscopy in art and in forensic science, really nice fields. And since 2012, she is the senior scientist and she works in the experimental part of the research group on surface spectroscopies and photonic or surface plasmon. This is really amazing background and curricula.
Thank you.
Well, and with this really nice scientist, we are going to start with the presentation of Mr. Alessandro Feis. And yes, you can, Professor Feis, you can share your presentation now to share your information with all of friends of the CAFÉ CON. And what is that? Because it's really amazing, the word photoacoustic effect. I think photoacoustic, I never listened before that. And then application in biology and medicine. Sure, this could be a nice topic to talk this afternoon, Professor Feis. Can you tell us about that?
Thank you, Laura. Thanks a lot. So just let me, before I start talking, acknowledge the Real Sociedad, Económica, Extremeña, the Amigos del País for the invitation. And let me acknowledge your nice help in talking about photoacoustic effect in biology and medicine. And last thing, not least, I would acknowledge your kindness to let me speak English, because I would like to speak Spanish in this talk, but it would be too difficult to me. So English,
no problem,
is a little bit easier to me. So
okay,
about the photoacoustic effect, yes?
Yes, when was discovered this photoacoustic effect? Really is as a novel effect, or this effect was there but we don't know, or we forget that this was the photoacoustic effect? When was discovered?
Yes, let's start from history. And the photoacoustic effect could be viewed as a historical physical effect, because it was already described in the 19th century, actually by A.G. Bell, who was the discoverer of the telephone. But the photoacoustic effect was discovered in the context of the photophone, which was one of the many inventions by Bell himself. And the effect was that he realized that if you had a beam of light and you could modulate in time this light beam, what we indicate today as chopping this light, then you could generate acoustic waves that you could really detect as sound. So you could modulate light and listen to modulated sound, which you may imagine could be applied to telecommunication. This was the idea, and this picture gives a bit the flavor of this kind of photoacoustic effect in a classical way. Nowadays, after so large time, we do not use modulated light beams, but we use lasers, which are often pulsed light sources. And I wish to stress that the laser was an invention which has the same age as myself, because it was presented in 1960, the year when I was born. And we ourselves in our group use laser light to measure photoacoustic effects.
But you are chemist, in particular, a physical chemist. How would you describe this photoacoustic effect from the point of view of the, for example, the molecules? Because there's a thing you can explain us about this photoacoustic effect from this point of view.
Yeah, thank you, Laura. This is a way of describing the photoacoustic effect and its consequences. And I will try to give a general view of the photoacoustic effect from the point of view of molecules, because, you know, there are many ways to look at reality, but for us, the chemists, and in particular the physical chemists, reality is made up of molecules. And then we tend to give a description of the materials and of the material world as made of molecules. And in this way, you can describe the photoacoustic effect, which is an effect which is started by absorption of light. So the first event is always absorption of light, which from the microscopic point of view is photons, little light particles. And this absorption is done by the molecules themselves, which build a material. And in this picture, which I sketched, you see these arrows on the left, which represent light. And on the right side, there are less arrows, meaning that a part of the light is absorbed. But who absorbed this light, making it weaker? Molecules. And in this picture, the material is made of these small stars. And you see some of these stars have changed their color because they have interacted with light, absorbing it. And in fact, from the point of view molecules, something has happened. They have absorbed light, but what does this mean? This means that they have exchanged energy with light. So light and molecules interact when light is absorbed. And from the point of view of molecules, molecules are changed by light, meaning that their energy content has increased, which we like to represent with schemes like this here on the right side. We sketch energy levels. These are these lines. And the molecules have changed in that their energy content is increased after absorbing light. So each molecule which has absorbed light has more energy content now. What happens now? Molecules have more energy, meaning it has been transferred from light to molecules. Now, energy must be transferred back from the molecules to the surrounding. And you should pay attention to this because this is what actually generates the photoacoustic effect. The question now is how energy can be transferred back to the environment. And I propose this. I would propose it the same way to students. How can you transfer energy back to the environment? And these are three possibilities: by emission of light, by external collisions, or by internal processes. Well, the answers to these multiple choice questions would always be correct because all processes may happen, but only this here, which is written in red, by external collisions, which technically is an intermolecular process, this is the process which leads to the photoacoustic effect. Because in this process, you have an energy transfer back from the molecules to the environment, and this generates heat, and this heat here is what generates the photoacoustic signal.
But it's really nice, but what do you measure this really little, oh, sorry, can you hear me?
Now I can hear. I missed you for a second, yeah.
Ah, okay. And now you can continue, or because I wanted to ask you, what do you measure in your laboratory? Because really it's amazing this effect.
Fine. So when you have this local heating I was telling about, then a part of the material will expand and generate a pressure wave. So what we measure is a pressure wave, and how can we do it? I will show you this picture, which was also in the invitation movie, which is taken by my laboratory. So on the left side, you see this picture, and you see a small part of liquid, which is this blue thing here, which would be a sample in my laboratory. When light comes here and is absorbed, then pressure can be generated if light is absorbed. It can be detected by what is called a piezoelectric transducer. And the piezoelectric transducer in this picture is this small object here, where my hand is just right now, which is close to the liquid I want to listen to. And the transducer detects pressure and transforms it, transduces it to an electrical current, which goes into this electrical wire. So in the end, what we measure in the laboratory is an electrical current in time. And this is a graphical representation of current, which is related to a very small voltage in millivolt, in time, which is a short time, which in this scale is microseconds, which is 1/1,000,000th of a second. So the transducer gives me current in time as an effect of a pressure wave, which is an effect of light absorption as the first event. So now imagine, just to connect what we see in a laboratory with real life, imagine that you substitute an ensemble of molecules like what we just have seen with an ensemble of small objects like real samples, for instance, biological samples or biological tissues or living animals are, then you will have a multiplicity of photoacoustic signals, all coming from different objects. And this is the meaning of my next sketch. When you see light coming in from the left side, going to a collection of many different objects, which are these objects with different shapes in the middle, and all of them generate different photoacoustic signals. So this is what we also could measure in laboratories. I do not measure macroscopic objects, but I could do it. And people who exploit the photoacoustic effect for more sophisticated investigations measure the photoacoustic signals generated by small objects. The important point is that the distribution in space of the absorbers can give information about the distribution in space of these absorbers. In other words, an image of these objects can be reconstructed by measuring photoacoustic signals. I hope I answered to your question, Laura.
Yes, yes, I think, and this is really amazing. That how do, because if you have the sample and you have how this little samples in the laboratory has the different information on the different sound, because it's acoustic, I think you can apply this method in biology and medicine. I can imagine a lot of fields that you can use this amazing effect.
Right. In fact, you can think that if you have a mean to construct or reconstruct, as we will see, images of objects, then you can apply this to obtain images of biological samples, of medical samples as well. And I would like to make a short presentation giving examples of photoacoustic images which can be encountered in biology and medicine, and maybe present you some possible perspectives, that is, future applications, if we are lucky, of photoacoustics in biology and medicine. So the principle is that I already told you that different photoacoustic signals give you information about the disposition in the three-dimensional space of different objects. To get the distribution in space of these objects, you will have to surround this ensemble of objects by several piezoelectric transducers in the space. And how you reconstruct the image can just be sketched. I myself do not make photoacoustic image, but I hope I can communicate you the flavor, which is, if you have a three-dimensional disposition, what we call an array of detectors around a very simple sample, like an illuminated cylinder, then what you get is a three-dimensional image of the illuminated cylinder. But if you have more, like in this example, I prepared a small piece of meat, now it is different, because when light comes in, then there will be volumes where light is absorbed, like the red ones, and there will be volumes where light is not absorbed, like the white ones. Now, if you can go back to the volumes which generate the photoacoustic signals, you will be able to reconstruct the three-dimensional image of this piece of meat. And this was a very simple example. But now I will give you something more complicated, like a true microscopic image, so an application of photoacoustic microscopy in this case, which means that I can measure micrographs by means of the photoacoustic effect. And I will tell you what these three images are. These are photoacoustic images, all three of them. And you see that you have more intensity, which, as I told you, means more absorption when the color is yellow. And you have less intensity where the color is black, and something in between when it is red. So you have more absorption when you have yellow. And these three here are biological structures, meaning that these are micrographies or biological images obtained through the photoacoustic effect. The objects giving rise to these micrographs are parts of a living rabbit. And the left micrograph is an ear of the rabbit. The middle one is the iris, so the middle of the eye of the rabbit. And on the right-hand side, you have the brain of a living rabbit. So the living rabbit is imaged by means of photoacoustics, which means that it is illuminated by light, and the piezoelectric transducers, when properly analyzed, give you back the image of the illuminated organ, ear, eye, or brain of this rabbit, which is alive and is fine because light does not disturb it too much. In these images, what is most apparent are the blood vessels, so the very small veins or very small arteries of these organs. So you mainly see blood through light absorption. But there is more, if I can go on.
Yes, yes.
You are not restricted to animals. You can apply photoacoustic microscopy to plants. And in these micrographs, you see different kinds of leaves, of plant leaves. And you see that these are 4 different plant leaves of different species, which give you different microscopies by means of photoacoustics. You know that, of course, that plants do not have blood. So what you are imaging now are pigments of plants like chlorophyll and similar substances. So you can think that you can apply photoacoustic microscopy to botanical studies, so to plant investigations. Then, a special advantage of photoacoustics is that you can do more than microscopy. You can measure functional microscopy. You can link biological structure to biological function. And this link is very interesting to biologists and to medical doctors as well, because you can associate the structure which you image to the function, and especially the biological function of that structure. And a natural way, like in this micrograph, is measuring a property which has to do with light absorption and with the function of the biological structure. In this example, it is the oxygen saturation. Maybe you are familiar with the measurement of oxygen saturation because we do it something at home. We measure oxygen saturation with a home saturimeter to measure how much oxygen content your blood has, because it is important for doctors. And in this image, you see oxygen saturation, which is more when you see red, which is less when you see blue, in a particular tissue. But there is more. You can look at this functional property, which is oxygen saturation in time. And in fact, on top of this micrography, you see time in seconds, because by means of photoacoustic imaging, you can see how the function changes in time. So you have, in the end, three informations: the structure, the function, and the dynamics, meaning that you see how the function changes in time. Very briefly, I would like to show you this scheme, because it is what is behind functional microscopy. Behind it, there is simply different kinds of light absorption, which are connected to the function, like the oxygen content of blood and of the blood protein, which is hemoglobin, which maybe you know. The point is, at a particular light color, which is what we call the light wavelength, like this blue bar in the scheme, you, in some examples, have different absorption of light, which depends on the oxygen content of blood. And this explains that if you choose this light color or wavelength excitation, your sample will be more sensitive to light, generating more photoacoustic waves if it is more oxygenated. And this is the principle of photoacoustic functional microscopy. There is more. To give you more examples, what we looked at until now were photographs or micrographs, which are applications of microscopy, but you can exploit the photoacoustic effect, making photoacoustic tomography. And this is also a medical term which we are familiar with, because sometimes we have to take tomographies. We normally do it by X-rays, but in the future, maybe we will be able to perform photoacoustic tomography. Tomography is a way of looking at three-dimensional structures. So it is like building a series of micrographies in 3 dimensions. And in this picture, which I tried to sketch, you have a projection of a 3-dimensional structure, like this smiling face. And you can imagine that different volumes give rise to different photoacoustic images. And if you can reconstruct in 3 dimensions these images, then you will be able to reconstruct a photoacoustic tomography. And this is promising for this reason, that light is relatively attenuated by absorption by the biological tissues, by the biological samples, but sound is much less attenuated. And this is promising because if you can detect light absorption by pressure or by ultrasound, then the signal which has to do with this pressure is not so much attenuated by the living tissues or by the living animals. And so you will possibly be able to measure photoacoustic tomography in the future with good efficiency. So now, if there is time, I will give you a few examples of photoacoustic tomography, which is an experimental technique now, but it is likely to become a commercial way of measuring diagnostic tomographies in the future. And these are two possible advantages of photoacoustic tomography. The first one, which is the photograph on the left, is that you do not need contrast agents because the light which gives rise to photoacoustic tomography is absorbed by tissues themselves. For instance, it is absorbed by blood. And the other advantage is that you do not need X-rays. You need ordinary light. You will take care that light is not too much, that it is not ultraviolet, but it is visible light or infrared and not too much of it. So in principle, photoacoustic tomography can be a less invasive way of measuring tomographies than other ways. And I will give you just two examples of photoacoustic tomography. This here is a series of three-dimensional photographies which can be done to tomographies, which represent an epileptic attack in a living mouse. And you see that there is a time scale in seconds, 25, 35, 45, 65 seconds. And you can see the epileptic wave spreading in the brain of this living mouse. The epileptic attack is triggered by an injection in this point where the arrow is. And you can see these yellow and red spots, which becomes more in certain areas of the brain. So this spreading of these areas in the brain corresponds to the epileptic attack. And this technique is interesting, of course, to biologists and to medical doctors studying epilepsy. And it is a promising method in biology and medicine based on photoacoustics. You can imagine more than blood. In this last example, you have an imaging based on fats instead of blood, and the images are images of our arteries, so our blood vessels. To facilitate imaging, you have the superposition of photoacoustic tomography with ultrasound images, which are known as echographies. So this is the superposition of ultrasound with photoacoustic tomographies based on different kinds of fats. And you know that this is important because fats in artery walls are something which are at the base of medical diagnosis for people suffering from atherosclerotic deposits in their blood vessels. And so for this reason, too, photoacoustics can be applied to perform tomographies of our arteries. So I think I will stop here with my examples, Laura. And
okay.
I think if you have more questions, please ask. If other people have more questions, I will be glad to answer.
Okay, thank you very much, Professor Feis. I think our viewers will be delighted with your clear and interesting explanation. I like a lot your pictures because they're really amazing. to see the inner of the eye of a mouse or the brain of a mouse, a living mouse, because it is, I think, this is the point, most interesting point. And overall, all these important application in the diagnosis on functional problems and maybe in the study of the structure and composition of the tissues. I think all the doctors and biologists, they will be amazed with this new. But now we are going to give way to the questions of our members, our friends in Brazil, Chile, Madrid, because sure they will have a lot of questions for you. And maybe we can start with the Professor Natalia Inostroza in Chile. What I think is really amazing. What do you think about this effect?
This was a really amazing talk. Thank you very much. I learned a lot. So, I mean, I have a lot of questions, but I mean, I think I had to be conscious about the times. So if you were thinking in the medical application, right? We can measure blood, tissues, and also maybe a tumor, right? How can you be sure that the absorption of this material will be enough to get a signal? I mean, I'm not sure about that.
That's a very, very good point, Natalia. Thank you. That's a very good point. How can we be sure? Ask the chemists. I would say, I do not want to put a competition between physicists and chemists, but this is really something which is linked to the chemical properties of your material. Mm-hmm. So, in the first point, you should, well, first of all, you should ask the biologists, because they must tell us which structures are more interesting to study. Mm-hmm. Then we should decompose them, so to say, into their chemical components and take, I would say, first of all, extinction spectra of the different components and be sure that light is absorbed at the wavelength which you are interested in, or at the wavelength which you have, because as you know, as a physicist, we do not always have all the wavelengths which we would like, and see which cellular components will efficiently absorb light. After checking this, you will experiment.
Okay, so is there any optical window that you have to select for your experiment finally?
Right. This is an extremely good point, because our bodies absorb basically at all the wavelengths. So we should select a wavelength window for the public, colors which are not too much absorbed by our body, so that light comes in. And naturally, from the point of view of nature, this is the infrared, because our body is more transparent in certain parts of the infrared light. And in fact, Natalia, this is the point. People are trying more and more to perform photoacoustics by infrared excitation, which is not so easy, but can be done, because then you will not always image blood, but you can. If you cannot, you will use contrast agents. In my talk, I said you do not need contrast agents, but sometimes you may want to employ them. You will project and design a photoacoustic contrast agent by infrared excitation and then use it. So the end answer to your nice question was, well, we will check if we can directly use photoacoustics, if we cannot, we will ask our colleagues to design a contrast agent.
Okay, great. Thank you very
Okay,
much.
thank you. Now I think Leonardo Negri Farini could ask something to our nice
First,
Alessandro Feis. Yes.
very interesting talk, Alessandro. I have a question about when you work with laser source, we have a problem to burn the sample, no? And how the temperature can be a problem in organic systems?
Good question. It can always be a problem, not only temperature, but in general, light irradiation. As you know, you should take care. You should pay a lot of attention when you use ultraviolet light. Well, there are standards. In fact, medical doctors know how much light you can use if you are exposing a human being to light irradiation at different wavelengths. You have definite values for infrared, for visible light, for ultraviolet. You should not exceed what we call the fluence or power density, depending on the quantity you're measuring, to avoid damaging your tissue. But the good news is that at reasonable light levels, so for low light levels, you can generate very good photoacoustic signals. On the other hand, our limit is that if light is too low, it will not reach tissues which are deep inside us. But to give you an idea, and to give an idea to the whole audience, nowadays you can perform photoacoustic tomography in living beings, like human beings, at the depth of 1 centimeter, more or less. Maybe something more, maybe something less.
It's too much, no? 1 centimeter.
It is good. For instance, you could do photoacoustic tomography of a hand. And in fact, this is what people show. Yes?
And no, just one more question, very quickly. When you saw the images from the inside of the body, the brain, I think,
Yeah.
there is a probe to put the laser inside the brain?
No, in that case, it was blood. So it was a direct image, so to say. But you have to use very often infrared light, because it goes through the bones of our head, so through our skull. And but we can start. We can start with this, yeah, and not use direct photoacoustic tomography. You can resort to contrast agents. Yeah.
Okay.
So there is also some line of research in that direction, trying contrast agents in the infrared, which are harmless. But there are some. And of course, many of you possibly already encountered metal nanoparticles and non-metal nanoparticles. They are also good candidates as contrast agents. Yeah.
Nice.
And I can imagine, as following with this matter, can it radiate from within the body? You can create a nanotomograph and take a pill or something, and you can create these images inside, from inside. Because I think 1 centimeter is okay, but maybe you can, I don't know, I imagine, but could be possible to take or irradiate this light inside the body and you get in deeper in the body. Is possible or not?
It is difficult for the brain, for the human brain.
No, yes.
Because
yes.
we have a lot.
No, but I was thinking maybe blood
but,
or heart. Yes. Yeah,
exactly. This image of the artery is something which actually we already do with other means. When we make a coronarography, for instance, we take diagnostics from inside the body. So in the future, surely it can be thought that we will be able to make photoacoustic tomography from the inside. So bringing light into our veins or arteries and reconstructing the image of the illuminated veins. Yes, it is something for the future.
Okay, thank you. And now we are going to listen the question of Mari Vega. Please, Mari Vega, sure, this is I have a lot of questions. Sure, you have one very interesting for Alessandro. We can listen. Sorry, Mari Vega. Yes.
Sorry. No, I was saying that I had a question similar to what you already asked, because I'm interested in the cardiological studies, as Alessandro said about the coronarography. I don't know how to pronounce it properly. When you do the photoacoustics, the tomography, when you show the arteries where the fat was located, how do you do these studies? Because it's not possible to do it from the outside of the body.
Correct. They tried both, if I remember correctly. So because
the image was just in a vessel, right, with the fat and with the coronary artery. So yeah. I was wondering how it was taken.
If I remember it correctly, they made the study both on the animal, because it was the animal there,
yeah,
and on the tissue. So on the isolated vein.
Okay.
And if I remember correctly, they compared both kinds of photoacoustic tomographic images from the isolated vein,
yeah,
and from the animal part. But they had to illuminate from inside,
yeah,
and place the piezoelectric transducers in a smart way, which I could not describe because I do not deal with imaging. Okay. But I know that there is a huge amount of different transducer dispositions. Just like a last hint, because I remember having thought about this. A piezoelectric transducer is a device which is commercial, and it is done for engineers normally. And it costs about only less than 1,000 euro for a single transducer. Okay. So you can imagine that you can easily place tens or even hundreds of transducers, yeah, to build a new device or instrument for photoacoustic tomography. I thought it was also the correct context, since we are at an economical society, yeah, to remember the economical aspect, because really photoacoustics in general is not so expensive. And this is also an interesting side for the future, because building new medical devices which exploit this piezoelectric transducer could also be an economically rewarding subject.
Yeah.
I think.
Okay. Thank you very much.
Mrs. Natalia, do you want to ask something else?
Not for me. It's okay.
Okay.
Everything is clear.
And maybe Leonardo have a question?
Yeah, I have one more about nanoparticles. I saw it's common now to use nanoparticles to produce heat close to tissue, and then you know it's possible to make a commercial product or in the future. I mean, to use nanoparticles in photoacoustic effect.
Yes, there are. In fact, there are several photoacoustic studies using nanoparticles to generate heat and building photoacoustic images in general on the basis of the heat which is released by the illuminated nanoparticles. And this is closer to the subjects which I study in the laboratory, because as you know, not all the nanoparticles absorb light at certain wavelengths to the same extent, because there are more efficient and less efficient. And this efficiency has a lot to do with the metal you use, the shape, the size of the nanoparticle, and so on. It occurs to me, if I can show one more slide. May I, Laura?
Yes, please.
But I did not show the last slide, I think, because the last one is actually a mixed image where you see the photoacoustic image red, which is the image of blood, and the green one, which corresponds in this case to an artificial tumor, which you can image on the basis of a biotechnological agent, but it also could be a technological contrast agent based on nanoparticles. In this case, it was a luminescent protein, but it might well be an ensemble of metal nanoparticles, which are also very efficient. The only thing, you must be sure that they are safe to us, which is also the question in nanotechnology and in nanotoxicology. So,
in that case, you need to put a probe close to nanoparticle? No.
In that case, you could directly image the nanoparticle.
Directly.
So you irradiate, the nanoparticle absorbs light, if it can, and it gives rise to the photoacoustic wave.
Okay. Thanks.
Okay. Mr. Maria Vega, do you want to ask something? Or because I have one question. I can imagine another interesting application is the possibility maybe to study the functions of the organs, because you say the functional application, but how, for example, the chemical composition of the substance are changing or are evolution inside our organ, or for example, our organ that is working properly, or our organ that is not working properly. And for example, maybe studying the secretion of a specific gland. You can compare the changes or the images of I don't know, every organ that are working. If you get a picture of this organ or an image of this organ, and then you can compare with an organ that is not functioning or something. It could be possible with a living human being. That it says, I'm going to the doctor, and you can observe inside of my body how my organs are working. It could be possible.
Sure. It will our hope, everybody's hope, is that it will work in the future. I can tell you about half, maybe, of the photoacoustic studies I found about functional tomography are looking at the blood flux, which is changing in time by means of photoacoustics. And that's good already, because there is a lot of information comparing healthy tissues and ill tissues, for instance, and so on. But in my opinion, at least for the future, the most interesting feature might be in neuroscience, exploiting photoacoustics to look at the nervous system and how it changes in time. Because, well, also in our brain, there is blood changing, and this is the same application I was mentioning in neuroscience. But possibly, we will be able to look at the propagation of the neural signal, of the brain signals by means of photoacoustics. This is not actual yet, not yet, but you know that we are always working for the future, at least in our hopes. So maybe in the future, the function which will be interesting for photoacoustics will be the neurological function. Who knows?
Okay. I don't know if some of you have another question. I have to say that our chat, all the people in the YouTube are not doing questions, but if one of you have another one, whatever.
Yeah, I have another question.
Okay.
Because I was wondering what's the differences between, for example, the echocardiography and the photoacoustic tomography, because you can, for example, when you want to see how the blood moves inside the arteries, you can see it through the echocardiography, as you just said. What are the differences?
That's a very good point, Maribega. In fact, for this reason, we often make a superposition, an overlay of the ultrasound image and the photoacoustic image. But where is the difference, as you asked? You know that ultrasound is sound in, sound out, whereas photoacoustics is light in, sound out. So what is the advantage of using light in the end? The advantage is that ultrasound images only so to say, they are important, of course, but basically give you information about the shape and how the shape changes in time. Photoacoustics goes a step beyond, because it can do selection by means of the light wavelength. So it can make a difference between colors. So red excitation, blue excitation, UV excitation, always listening to the response. So photoacoustic tomography in medicine has the potential of making, of being more selective in a way. So this is our hope. Okay,
thank you.
And I suppose that now the greatest effort will be studying the images of these samples in the laboratory, to have a wide and extensive catalog of images to say, oh yes, if I want to see what kind of molecules or substance I have, the pattern to compare. And I think the other topic need to be to design this photoacoustic tomograph to have the 3D images more accurate, and the software, because I think you get the images and then you have to build the 3D images like with a computer. And I think that could be a good relation between engineers and informatics, and maybe a lot of people working together to have this new techniques, I think, and doctors, sure, doctors in medicine, that they will suggest what they need exactly, or what they are looking for. Is true or not?
That's correct. No, that's 100% true. Especially this role of engineers is particularly important because they tend to develop new methods, which are really practical methods, no? To aid people in reconstructing photoacoustic tomographies. We, the scientists, have a somewhat different approach because we tend to look at the basics and see how molecules work, see how photoacoustic signals are generated and how and under which conditions and more. But the engineers will really look for the link between images of the real world and improved methods. It's amazing. And of course, medical doctors in the interpretation. You know, Laura, it's amazing to ourselves too. I was impressed by the development of biological and biomedical photoacoustics because I didn't know. I knew photoacoustics as a means to study photochemistry, which is a completely specialistic area, so to say. But nowadays, people are investigating in the development of new instrumentation, new diagnostic tools, new ways and faster ways to diagnostic illnesses, for instances.
And imagine that the result of the photoacoustic scans could be an hologram. Imagine that you can see the 3D, will be maybe science fiction, but I like to imagine the future. And imagine this photoacoustic tomography represented by holograms, 3D, with different colors, maybe, I think we need to, I think next century, or maybe not. But I think it's really, really amazing. I don't know if all the things we can imagine, we can really, at the end, we can have people to work in that new ideas, because we need more people studying in the universities, physics, chemistry, engineering. Yes, I think this, well, we work in La Económica, we are working to get the science all accessible for the young people overall, maybe in English for the overall the world, because we want to encourage the young people to study science and these amazing fields. Imagine the possibilities we have in medicine or in botanic or in maybe other fields in the science. Okay, I think if you want to do one last question or one last comment to Alessandro, because I think it's nice time to finish. Maybe one last comment to Alessandro, because it's time to finish our lecture. Maybe Natalia from this nice place in Chile.
Alessandro, have you ever tried in your experiment using different contrasts, right?
Sorry, Natalia?
In your laboratory, have you already used different contrasts? For example, molecular dyes?
In my laboratory, we have started substituting, so to say, molecules with nanoparticles.
Oh, okay. It's better? Which is the advantage? I'm not sure.
The advantage is that nowadays we can design nanoparticles so that they will absorb light where you want, I mean, at the right wavelength, and be stable. Because some dyes are very nice, absorb where you want, but after some light pulses, they are destroyed.
Ah, yeah, right.
Nanoparticles are often more stable, and this is the advantage. Besides
this is for the future now, then. No? So now you are focused just on nanoparticle. Okay.
Yep.
That's great.
Thank
you very much.
Now, for example, Maribega, you want to say something?
Yeah, regarding these nanoparticles, are you saying only like metal nanoparticles or semiconductor nanoparticles, or what kind of nanoparticles do you want to use or you are already used?
Nice question, Maribega, because you know from our collaboration that we have always liked silver nanoparticles because they are active in Raman. But that was exactly the reason why I started doing photoacoustics on metal nanoparticles. I selected gold. But nowadays, the interesting thing is using non-metallic nanoparticles, for instance, sulfides or semiconductors of different kinds, if they absorb light efficiently. So technically said, if they are plasmonic nanoparticles. And there are a lot. This is a completely new branch of photoacoustics.
It's also interesting. Yeah, there are also some semiconductor nanoparticles that are being used in for CERS.
Correct.
Already, yes.
Maybe in the future. Maybe in the future.
I think we have to repeat this conference maybe then later 3, 4, 10 years, and we can see what is the final of the film.
Right.
the end of the film. Okay, now Leonardo, please, can you say something to Alessandro, please?
Yeah, last question about the photoacoustic. It's a contact, no? You should be the detector in contact with the sample.
Right.
It's a problem to make 3D images.
There are two ways. Some people place the transducer directly on the tissue or animal, whatever, or many transducers, and then they move the transducer or they move the animal, and the transducer is in contact with it. Or you, if possible, you immerse the animal in water, for instance, and water will ensure the contact between the transducer and the animal. Paying attention, of course, you can do it. You can do it.
Very interesting.
As you wish. But you will have to take care. Not only for the animal, but also for the transducers. No? You have to place the
of course. Correct. It's still breathing. It's so far so good.
Yeah, exactly.
Subaquatic transducer. You got to work. Okay. Well, I want to say thank you to all our friends in this little community of CAFÉ CON. I'm sure that you will be amazed with the following lecture we are working for this new season. I hope that you enjoy this conference because I learn a lot. Every time I learn these really amazing different topics, and overall with these really nice scientists, because I think people can read a report or an article in a magazine, but I like to see the face, the people were speaking in live, and know how our scientists look, because sometimes we know how is a football player or artist, a singer, but we never know the face of our scientists. And this is for me very important to have your faces and your smiles and your words directly in the CAFÉ CON. And I want to say goodbye because it's late. And thank you to all our viewers and our friends for CAFÉ CON. And we wait in October with another really nice lecture. Thank you to all of you.
Thank you.
Kisses.
Thank you.
Bye. Ciao.
Thank you for this invitation. And of course, we are not the nerds, so we are normal people working in a passionate way. So please keep this kind of activity for outreach. So important.
Oh, thank you so much. And this next, I hope that, well, I send you the mug. But, yes, but Brazil and Chile is really far. I don't know when they arrive, but I'm sure this little present will arrive to your house. Okay.
Okay, great to know that.
Yes. Okay. This is from Spain.
Okay, ciao.
Okay, thank you very much.
Ciao.
Ciao.
Bye.
Nice to meet you.